Welcome, everyone, to the May 3rd meeting 2022 of the Oregon Archaeological Society. I want to hand off the introduction of Dr. April Nowell to Dan Stuber, who will tell us all about April and what her talk's going to be. Dan, take it away.
Well, thank you very much, Jim. Hello, everyone that are sitting there in the auditorium at OMSI, and to all you Zoomers at home. You're in for a special treat tonight, and I have the special honor of introducing our speaker, my colleague and very dear friend, Dr. April Nowell. Dr. Nowell is a Paleolithic archaeologist and professor of anthropology at the University of Victoria in British Columbia. She has an outstanding career, so varied that I won't even get to it all here, but she is a researcher, a professor, an author, and presenter. She is widely published and has co-edited volumes including Stone Tools and the Evolution of Human Cognition, Archaeology of Night, Life After Dark in the Ancient World, and the forthcoming Culturing the Body, Prehistoric Perspectives on Identity and Sociality. She is also the author of the new book, Growing Up in the Ice Age, Fossil and Archaeological Evidence of the Lived Lives of Pleistocene Children. And I gotta highly recommend it. It's a fascinating read, and it's got the greatest cover. Dr. Nowell directs an international team of scientists studying lower and middle Paleolithic sites in Jordan. In 2016, she and her colleagues discovered the world's oldest blood residue ever detected on stone tools, showing that 250,000 years ago, hominins ate everything from ducks to rhinos. This research was published in the Journal of Archaeological Sciences and was named one of Time Magazine's top 100 discoveries of the year. Dr. Nowell's presentation this evening is Getting Blood from a Stone, Excavations at a Paleolithic Oasis in Jordan. Please welcome Dr. April Nowell.
Thank you so much, Dan.
You're welcome, April. So good to see you here.
Yeah, thank you. You too. It's so special for me to have you right there. It's awesome. So it's an absolute, yeah.
Well, I'll mute my face and listen intently. Thank you.
See you later.
See you later.
Okay, I'm going to share my screen now for everybody. There we go. And from beginning. And I'm just going to make this a little smaller. Okay, there. Okay, excellent. All right, well, thank you all so much for inviting me here this evening. I really wish I was there in person with you. We've postponed this talk a couple of times because we kept thinking it would be possible for me to come down and visit all of you in person, but that wasn't possible, unfortunately. So I'll have to find another excuse to come to Portland in the fall to see Dan and other friends. Okay, so I'd like to get started. Here's a picture of where I work with Dan Stuber, actually, in Azraq, Northeast Jordan. And Azraq is the Arabic word for blue, and it's from where we derive the English word azure. And so with that in mind, there's something incongruous about the name of the town and then the feel of walking through this very dry, dusty, trucking town of Azraq in Northeast Jordan. But if you can imagine, and I'll just this is what it looks like today. But this is what it looked like not too long ago. But millennia ago, before Lawrence of Arabia planned his attack on Damascus from this castle, they sometimes call it Lawrence's Castle that you see there on your left. And before the Crusaders built their castles here, because right now when you look at the desert, you would think, why would people build castles in the middle of the desert? And before the Romans erected forts here, in fact, some of the walls around the back of this castle are Roman walls. So before all of that happened, Azraq was this lush oasis teeming with life. And in fact, some of the people living in Azraq today still remember as kids going fishing and swimming in the lakes and ponds in the town that have now since disappeared. So my talk today is really a story of human resilience in the face of climate change, and a treatise of the shifting relationships between plants and animals and people in a fragile ecosystem over thousands and thousands of years. And it's really, given our current fears about climate change and the current challenges that climate change poses, it's really a story that I think has relevance for the world that we live in today. So just to kind of set the story or give you the context here, Azraq is part of what we call the Levant, so a region of the world we call the Levant, which is an area that includes the modern day countries of Jordan and Israel and Syria and Lebanon. And as a paleoanthropologist, it's one of the most exciting regions for me to be working in, because these countries formed what we call the Levantine Corridor. And it's a region or an area that connects the continents of Africa and Eurasia. And so for thousands of years, plants and animals, including hominines is the word that we use for humans and their extinct ancestors. So plants and animals, and including hominines, moved back and forth along this Levantine corridor. And in fact, a colleague of mine refers to it, because of the sort of busyness over the millennia, he referred to it as a Paleolithic bus station, because people are always coming in and out of it. And actually, and I'm sort of reminded of the movement even of plants between these two regions when I work in one part of Jordan, not the part I'm going to talk about tonight, but another part closer to the Dead Sea. And you see a lot of African flora. You see baobab trees, like from the story of the Little Prince down there. And again, that shows you, that kind of emphasizes that connection between the continents. And so Azraq itself is actually also at a crossroads. So not only is it part of this Levantine corridor, but the city of Azraq itself, as you can see on the right-hand side of the slide, and you can see the little orange dot here, is really also, at a crossroads itself between, so it connects Jordan with Syria and Iraq and Saudi Arabia. And so there's a good reason for why it's a trucking town. There's always trucks barreling down the highways there, carrying fuel up from Saudi Arabia, and large stones and other kinds of goods back and forth. And they often pay young unmarried men to drive these fuel trucks, these oil trucks and so on, because it's fairly dangerous to do. And they're the guys who are crazy enough, I guess, to do a lot of that, or desperate enough, I guess, is the other side of that, maybe, to do that kind of job. So very, as I say, dry, dusty place with lots and lots of trucks coming through. So it's still, in some senses, a bus station, as it was way back in the Paleolithic. And so the actual site that I'm going to talk about is part of, it's situated within the Azraq Wetland Reserve. So the town of Azraq itself is situated within a larger region that's called the Greater Azraq Oasis area. And it was once a really large expanse of wetlands. And so again, for thousands of years, this oasis area was fed by two was populated by two spring-fed marshes. In the north, the one that we refer to as the Druze Marsh, and then in the south, the Shishan Marsh, and that's what I'm going to talk about today. And so due to climate change and overpumping of the marshes, the springs collapsed in the 1980s. And there's been an effort heralded by Jordan's Royal Society of Nature, the Society for the Conservation of Nature, to recover about 10% of the former extent of the Shishan Marsh. And that's what you're looking at here. And it's absolutely a stunning place to walk around. You've got these bridges, you've got bird blinds there, so you can watch migrating birds. They've reintroduced water buffalo, and they're starting to see a number of different kinds of birds and plants and animals coming back to this area. But as I say, it's only about 10% of what it once was. And the Druze Marsh was actually never restored. It's actually, that's where we started working in, I think around 2009, 2010, I can't quite remember now. And we've done some salvage archaeology there, but they were in the middle of making a children's park in that, what used to be that northern marsh, because it's just dry desert now. It's right across from that castle I was showing you at the very beginning. And so we were collecting as much data as we could, literally just a few feet ahead of the cement mixer. So there is now a latrine, a bathroom, I should say, on top of what is probably one of the oldest sites in Jordan. So it broke my heart a little, but at least the layers are preserved underneath that. So perhaps in the future, there'll be an opportunity to come back and re-excavate in that area. And so the other thing I wanted to say about that is the reason why, as I said, there's, you know, the climate change and overpumping and so on, is the reason why these marshes have mostly collapsed. And even with the 10% that's still around here in the Shishan Marsh, about 1 out of every 4 glasses of drinking water in the capital city of Amman comes from this marsh. And there are also approximately 500 illegal wells that continue to operate there. So people are putting in these illegal wells. At the same time, people need to have water for their families, for their animals, for farming and so on. So it's hard to argue with their need for water. And at the same time, it's also a very difficult battle or war to wage that the conservationists are trying to win, I guess, by keeping at least 10% of this marsh going. The only good side, I guess, of the dropping water table from an archaeological perspective is that it revealed stratified Paleolithic sites with just an abundance of stone tools and faunal remains or animal remains associated with both of these paleo marshes. And as I say, I'm going to focus just on the Shishan Marsh here. But as you can see, compared to this part of the marsh that's been restored, this is what the other 90% of the marsh looks like. And when you walk around here and you look at any sort of natural cuttings or places where they've had a bulldozer come in for one reason or another, you can see Paleolithic materials just pouring out of them. So they're really quite rich sites. So as I say, my talk this evening is going to focus on excavations in the Shishan Marsh that we began in around 2012. And it's what we call Shishan Marsh 1 or SM1 located in the Wetland Reserve. And based on our research, we know that hominines repeatedly occupied SM1 between 300,000 years ago and about 130,000 years ago during a period of climactic fluctuation that resulted in a transition from an exceptionally wet period to modern or even more arid conditions than what we see today. So we had a number of different questions, but really one of our primary questions was really a simple one. It was, how could hominines living 300,000 years ago survive and in fact seemingly flourish in an environment that's so challenging that people today have trouble surviving in? Excuse me. And so before I get into some of the things that we found, I just wanted to very briefly talk about the kinds of dating that we're using to date our sites. So many of you are familiar, or I would imagine all of you are familiar with dating techniques like carbon-14 dating. And certainly as Paleolithic archaeologists, we use carbon-14 dating for some of our sites. So rock art sites, for instance, the Ice Age art, cave art sites, if they have carbon in the pigment, we'll use C14. But for the kinds of things that we're doing in Shishan, carbon-14 just really isn't old, doesn't go back far enough in time. It really sort of peters out around 50,000. Some people think it's possible to extend it to 100,000. I get more and more nervous as you extend beyond 50,000. Maybe I'm a little conservative. So instead we use uranium series dating, and we also use optically stimulated luminescence dating, or OSL. And just to kind of briefly say what OSL does is basically that what we're trying to do is see when the soil that we're excavating, when the surface that we're excavating was last used as was last a surface that hominines could have walked on. And in order to collect our samples for OSL dating, we have to collect them in absolute darkness. And so here's one of our sites. This is not SM1. This is one that's close to the Dead Sea, and we're right by a road, so it's super dangerous to be able to be out there at night with no lights, and drivers in Jordan I'm from Quebec, and we're known for being bad drivers. But Jordanian drivers at a whole other level, and so it's a little dangerous to be on the side of the road in the dark there. So we take our samples during the day, and I'm laughing, and I'm with my colleague Mike Bisson, former chair of anthropology at McGill University. We're both laughing because until 2 seconds before this photo was taken, I was covered in thick plastic as I took a sample in the dark. Take something that's basically a PVC pipe, or looks like one, and hammer it into the sediment, and then wrap the ends with duct tape and all kinds of things like that in order to seal them. And it's really hard surface to work with, and so it takes a long time, and it's super hot under there. And we almost caused an accident as a bus taking a bunch of families down to the Dead Sea for a little weekend holiday. We're watching what we were doing, and all of a sudden this Canadian archaeologist pops out from under a bunch of plastic, and the driver was quite surprised to see that. And then of course we had the little bit of we had the difficulty then of getting our samples to our lab. We work with a lab in Oklahoma, and we also work with a lab in Germany, and to get the samples out without them opening them at the airport was a bit of a challenge. So I was trying to convince airlines to take what looks like a pipe bomb from the Middle East to Oklahoma. So, in the end, that wasn't without opening them, I mean, so that was a no-go. And so eventually we found ways of sending multiple samples and so on, and just hoping that some make it safely through. So adventures in doing Paleolithic archaeology. Anyway, so I just wanted to give you a little context for how we get our dates of 300,000 to 130,000. And so then the other question that so this is the other thing I wanted to do in terms of a background for you, is to also look at placing this in time and to look at what hominine species might have been responsible for making the stone tools at Shishan Marsh 1. And so as you can see, this is, I love this phylogeny here, this slide of our evolutionary, you would say evolutionary tree or family tree, but really when you look at it, you can see that maybe we should be using the term bush. It should be like a family bush in the sense there's so many branches. And in fact, I don't have all of the species on here. It's actually a few years out of date. So there are probably about 3 or 4 species that are missing, but it's such a great visual that I continue to use it. So we have evidence for hominines on the human line going back 6, 7, 8 million years ago. But the time period that I'm talking about today is just this little sliver of time within that orange rectangle. And as you can see from that little sliver of time, we have a number of different species that might be around in the Middle East at the time, including Neanderthals, something called Heidelbergensis, of course, modern humans, possibly Erectus. I'm going to talk about it more on the next slide. And Floresiensis, as the name implies, is in Flores and not where we are, not at our site. So when I used to give this talk, when I started giving an earlier version of it about 5 years ago, then my next 7 slides were all about Homo erectus. And it was such a nice, it was, because I love Homo erectus. They're, the middle child of evolution. They always get forgotten. And yet they're super cool, in the same way, the first ones to leave Africa and all kinds of interesting things about them. But with better dating and more recent discoveries and sort of analyses of things and so on, it's likely that Homo erectus probably disappeared from the Levant closer to 400 or 500,000 years ago. It's not impossible that they're still around because they are certainly in parts of Indonesia and so on. So even about 100,000, some people think to 50,000. I'm not so sure about that, but at least, well beyond the time frame that we're talking about, but probably not in our region. Neanderthals are certainly in Israel and surrounding areas, Iraq and so on. So it's not impossible that they were here at this time, certainly for the younger layers, but maybe all the way back. Modern humans could certainly be here for sure. And then we used to talk about Homo heidelbergensis, which is kind of a catch-all for a variety of what we call archaic sapiens at that time. There's been a new paper out suggesting we rename all of these fossils to Homo bodoensis. I'm not going to get into the controversy because it's not really germane to our talk today. I guess the main thing that I wanted to say is that there are many different possibilities for who could have made the stone tools and brought and hunted and so on there, and it could have even been a variety of species, so multiple species at different times using this site. So what I'm going to do instead of focusing in on one species is really to talk about what we know about the Shishan Marsh hominines in general. And so here's a picture that drawn by my colleague, Carlos Cordova, and he is the geoarchaeologist on our site and responsible for a lot of the dating and a lot of the sedimentary work, geomorphology work that we're engaged in. And so if we ask what life was like for hominines living at Shishan Marsh, We know that it was repeatedly occupied, as I said, by hominins between 130 and 300,000 years ago, as we have minor fluctuations in climate. And these fluctuations really change the extent and the distribution of water sources in the oases. So sometimes we talk about paleo marshes or paleo lakes even, and other times we're talking about seasonally available springs. And so the occupations that we've been able to uncover occurred during these warm and dry conditions associated with a transition, as I say, from a receding lake to these marshy ponds and even these springs. And we've worked quite a bit collecting information on the environment using phytoliths and diatoms and sponge spicules, which are kind of these structural elements of sponges. And we've collected those from our archaeological layers, and they indicate a predominance of aquatic vegetation. So, for example, the grasses, so things like reeds and rushes and cattails and sedges, among others. And then surrounded by what we would call a desert scrub. So these chenopod scrubs and sand dune kinds of vegetation. So very typical of hot deserts in the Middle East today. And then I just wanted to show you a slide here. This is from a brand new article that was just published by my colleagues, Kelsey Boyd, Chris Ames, and Carlos Cordova just last month. And this is based on Kelsey's master's research into the palynology at the site. So basically, if we look from older to younger, moving our way upward, we can see that the results show the presence of a perennial or near perennial water body in central Azraq Basin, at least more than 270,000 years ago, at a time when we have these very regionally wet conditions across the Levant. And then that's followed here in the middle by the development of an extensive marsh environment along the western margin of Shishan Marsh, associated with hominins that are making late Acheulean sort of hand axes and so on. We used to say that it has to be Homo erectus, but we now know that there are a variety of different species that make hand axes. And so this landscape was a more mosaic of terrestrial and aquatic grass communities, surrounded again by open grasslands. And then, of course, at the very top, we start to gradually transition into that regional desert scrub that I mentioned earlier. And so that wetland and associated grassland would have provided diverse and abundant resources in an otherwise very arid environment. So it probably acted as what we would call a desert refugium, so an oasis, in other words, for fauna and hominins during the middle to late Pleistocene transition. And we have colleagues who are working further south to us in Saudi Arabia. They have a project called Green Arabia, and where they're looking at those sort of refugium areas as well. And we have a colleague who was working in Syria until it became impossible to work in Syria. And again, there's sort of these tying of people to resources. So probably hominins were moving in and out of the region. They were probably very aware of where the next water source, this next oasis or refugium area was, and they probably moved there at different times during the year. So just to kind of look at very briefly at some of the animals that we have, at Shishan Marsh. The taxa are indicators of very dry, open steppe environment with some shrubs. And we know, of course, that the fauna would have been attracted to the water margins. This is the work of my colleague, James Bokinis at Boston University. He's our zooarch person. And so we have found camel, wild cattle. So the bones or faunal remains of camel, wild cattle, equids, so may have been some kind of steppe asp, for example, steppe rhino, extinct elephant and Asian elephant, and lion. And here are just some pictures of some of the remains. I have to show you that, I have to tell you that I'm kind of fudging a little and showing you the absolute best examples of fauna from this site. Most of our fauna is quite degraded, little fragments that are very difficult to be able to identify. Poor Jim, our zooarch person, tears his hair out, because we have such acidic soil, but it's actually better than what we had in the northern marsh, in the Druze Marsh. We found virtually nothing there. So we do have some preservation of faunal remains here. So here we have a wild cattle molar, we have some camel. So again, teeth and vertebrae, and then some other remains here. So limb bones and so on. Here we have the calcaneus. Some more of the equus, and here is again, this idea, maybe a step bass of some kind. Molars, and then we have what looks like remnants, tooth belonging to a lion, and then probably another large carnivore, possibly a spotted hyena. That's our best guess based on a variety of metrics. And then of course, extinct elephant. And I think this one, I think if it's not in a museum right now, will be part of a display. And then moving on to the stone tools, and this is the work that I do with Dan Stuber, and I'm so grateful to have him, to work with an amazing colleague, such deep knowledge, and yeah, just a real privilege to work with him. So Dan and I have been working on and off on these tools for quite a few years now. COVID has put a bit of a cramp in our style, in the sense that we haven't been able to get together to do the last little bit that we need to, but hopefully that's going to happen soon. But overall, we've been able to identify approximately 10,000 tools, and I use that word in the broadest sense to include, lithic artifacts, to include everything from, the tiniest chipping debris that you would produce in the making of a stone tool, all the way to some of these beautiful hand axes. A number of them are coming from local flint, so less than a kilometer away, and from local wadi gravels. Quite often, they're picking up small tools, so tools that are maybe a couple of centimeters in size, and we think they're using them for very specialized tasks. Dan's done some work looking at useware on these tools, and they're likely being used to process rhizomes and other parts of plant materials, maybe some small fish or other aquatic animals. And as Dan has reminded me in past occasions, there's nothing like a sharp blade for doing a whole variety of tasks. So those ones seem to be made from the local wadi gravels, but our hand axes often are being made off-site, from flint that's further away. And that work, sourcing some of that flint, is work that's been done by my former doctoral student and now Dr. Jeremy Beller for his dissertation. So these beautiful hand axes then made offsite, brought into SM1, and all of them have very sharp edges. That's something that Dan and I noticed, that there's very sharp edges, very little evidence of damage. I mean, it's not that there's no damage, but there's very minor damage on it. There's not a lot of evidence for the rolling or anything of them. So we don't think that these tools have moved around a whole lot. We think that they're pretty much in situ, pretty much we're finding them where they were used and discarded. So that's really important for us in terms of being able to make some interpretations of the site. And then now we come to what I think is the most exciting part of the research that we're doing, which is our blood residue testing. And in much the same way that genetics has revolutionized our understanding of the evolutionary relationships among hominins, paleoproteomics is set to transform how archaeologists study animal species and by extension, the paleo environment and subsistence strategies and all of that. So basically, paleoproteomics is a field of molecular science that relies on protein sequencing technology. One exciting method in zoo archaeology is zoo archaeology by mass spectrometry, or what we call zooms. And zooms is a scientific method that identifies animal species by means of characteristic, what we call peptide sequences in the protein collagen. And so it's a protein fiber basically found in the bone. And what it allows you to do is, if you take a tiny sample of this bone, it can give you 2 species or maybe family designations. And so what's really interesting for us about this is that we've been able to use zooms at our site. We've just started to do that recently as a complement to some of the blood residue work that we're doing. And it's allowing us to be able to confirm and augment our blood residue results, and also to expand our ability to be able to identify the faunal remains at our site, because as I said, most of them are just almost like bone splinters. And so we wouldn't be able to do that otherwise. There's been some really cool uses of zooms in other aspects of human evolution. I can talk about it in more detail, but used on Neanderthal sites, for instance, by colleagues of mine. But what I want to focus on is crossover immunoelectrophoresis, or CIEP. And this work is being done by Cam Walker, who I was hoping would be here tonight, but I don't think he is, but maybe he'll pop up and surprise me. And also, but I did see that we have John Fagan here from Archaeological Investigations Northwest, and John and his company have been very much involved in the blood residue testing for us, and so such a pleasure to see your name here tonight too, John. And so I'm going to give you a very basic explanation of CIEP that I am a little embarrassed to give with John here. So I will totally turn it over to him for more detailed explanations if any of you have any specific questions. But the way I often explain this technique is, well, first of all, I'll say that it's a technique that was first developed by the Royal Canadian Mounted Police, by the Mounties, and it was to be able to identify blood pools, basically in crime scenes. And it was developed about 50 years ago. And since then, it has become a very rigorous technique that is used in forensic contexts all around the world. I think it's also used sometimes in fishery contexts and things like that, when they need to identify a species from protein residue. And so the way I often explain this is to have people think about the relationship between an antigen and or a virus and an antibody. And when I first started giving this talk about 10 years ago, it wasn't, people were less familiar with talking about antibodies and viruses and all of that. And unfortunately, we're so we can't escape discussions of those things over the past 2 to 3 years now. But basically, when you think about when an antigen or some kind of foreign body, a virus, something like that, enters your system, enters your body, your immune system kicks into action and develops antibodies that target and hopefully eliminate that antigen, that virus, that foreign body, and so on. And so we use that. So that very simple, basic principle underlies CIEP. Basically, what we do is we extract, and when I say we, I mean Cam and John, extract residue from stone tools in order that we need to use for this technique. When you think about stone tools and you look at them, they look very opaque, they look solid, but actually when you look at them carefully and particularly under a microscope, you see that stone tools have multiple cracks and fissures in them and microfractures and all of those things. And so when they're used to cut and slice and scrape bones and meat and hides, it happens that blood and fatty tissue and soil particles are forced into those cracks. If you can just sort of think about the movement or the motion of the way those tools are being used. And so this combination of materials is very resistant to microbial attack. And if the fatty tissues develop into a type of fat, this posterior that's sort of looks like a waxy substance. If that develops and the tools take in calcium ions from the surrounding water or soil that maybe has a high mineral content to it, then this residue becomes nearly insoluble as well. And so even though we're not getting a lot of faunal remains preserving because our site soil is very acidic, we actually have this protein residue, this blood residue, but it's more than blood, it's all the tissues I mentioned as well, preserving really, really well. And so basically what we want to be able to do is figure out whose blood or fat or whatever, but that tissue, what kind of animal does it belong to? And so we're not doing ancient DNA, so we're not doing the same kinds of techniques, but coming back to that idea of antigens and antibodies, basically what we want to do is, if, for example, I want to know if my stone tool was used to butcher a horse, I would take a blood sample from a horse, distill it, so I'm taking just part of it that I need, and then I would inject that into a host, and usually we use goats, and these are the two friendly, happy goats that you see here, and we inject that into a goat, and we come back 2 to 3 months later, and we can do an extraction there. And in the meantime, a blood extraction, in the meantime, these animals have developed antibodies to target that foreign particle, the horse blood that we've injected there. And again, I realize that this is an oversimplified explanation, but I think it kind of gives you a good idea of how that works. I would also like to point out that we don't stress the animals unnecessarily. A lot of the work that we do is during regular veterinary care or checkups. And so, so then we have those antibodies, and then what we do then, or what's done in the lab, and again, simplifying this, is that we bring together that residue sample from the stone tool that we've taken, and we bring it together with this, what we call with antisera, that we've produced, or antiserum, put them together. And if there is a reaction, then we know in this particular case that we have horse blood or horse proteins on these stone tools. If we don't have a reaction, then we don't know what it is, and we have to try again. And so we go through a variety of antisera, testing them against a whole variety of different animals. And so what we were able to do, much to my surprise, was to find the oldest identifiable blood residue results in the world. And I have to give Dan credit for this, because we were working in my tiny lab up here in Victoria, and Dan said, you know, we should test some of these stone tools for blood residue. And I was like, oh, that's really, you know, it's not possible, because Dan's saying, oh, you know, my buddy Cam has had really good success with this technique. But I said, yes, but that's in a younger context. There's no way it would preserve as long on our stone tools. I think the next oldest examples we have are around 11,000 years old. So I thought there's no way this was going to work. But we decided to take a risk, and we chose 5 or 6 tools. I think we did 5, and then I added a 6th one at the very end, based on the amount of wear that we could see on these stone tools. So what ones look like they were just used to death, and, you know, would have the best possibility of having any results, you know, any residue surviving on them. And I remember I was on vacation and just got this email from John and Cam telling me that we had one hit, that we had a horse, and I could not believe it. And so very quickly, we decided to send a bunch more tools, and then we got more funding, this time from the Leakey Foundation, and also in partnership again with Archaeological Investigations Northwest. We're able to afford to do some more testing, so a larger number of tools and a larger number of antisera, including ones that had to be custom made. So it's relatively easy to get some of these in a forensic context, but some of them, such as rhino, we were not able to get. They don't end up in crime scenes that often, or at least not locally. So Cam had to do that one from scratch in-house. So yeah, we found the oldest identifiable blood protein in the world. We found, I'm going to just tell you the ones that we've published. So we found evidence on our stone tools that they were used to process camel, bovid, which is a wild cattle, lion, Asian elephant, duck, and rhino. We have a number of other ones that I can't share yet because we haven't published them yet, but I'm very excited to do that shortly. That's my goal for May and June, is to finish writing up these results. And so, and as I said, and as Dan said in his introduction, we got a lot of interest in this research worldwide, the New York Times, to Washington Post, to the Smithsonian, to being named, as I say, one of Time Magazine's top 100 discoveries for that year, so for 2016. And so for me, beyond the sort of cool factor of having the oldest here, is that there are a number of implications that we can take away from this research. So first of all, this research gives us the first direct evidence for how the stone tools were used. So we have use wear, we have cut marks on stone tools, and those are all great proxies for how these are being used. But the blood residue, or the protein residue, is really, you know, the first direct evidence of what these tools are being used for. So that's really exciting. And in many ways, they've confirmed the kinds of inferences or you know, suggestions that archaeologists, interpretations that archaeologists have made. The second thing that's really interesting is that we can see the broad spectrum diet that these hominins were eating. So 300,000 years ago, they're eating everything from duck to rhino. And we know that today people living in challenging environments will often eat anything and everything that they can get their hands on, right? Like, just it makes sense. And we can see around 120,000 years ago, that this was a strategy that hominins were using. They, very broad spectrum, but we didn't know how far back in time this went. And so these results give us a sense of the kinds of complex dietary strategies that these individuals were following. And what you can glean from that as well, or the extension that the inference we can make from that as well, is that it tells us a lot about their technology and their hunting strategies. So for example, the way you hunt a duck is very different from the way you hunt or scavenge a rhino. And you may be using different kinds of tools. So for example, I have colleagues who work in a site dating to 300,000 years ago in Germany, where they have these kinds of spears. Instead of having sort of one pointed edge, they have these two sort of these two edges, two business ends, shall we say, that when you throw it up in the air, it kind of creates these large, or cuts these large swaths in the air and can take down a number of different birds. And so they have those dating to 300 remnants of those kinds of hunting tools 300,000 years ago in Germany. They also have a lot of duck egg shells in Germany as well. So it's quite possible that they're using the similar kind of technology here at Shushan Marsh, but we don't have wood preserving here the way they have at some other sites. And so the technology might be different. It might also tell us something about the kinds of task groups that they're involved in or developing, because you don't want to be living right next to what is essentially an African watering hole. You don't want to have your little ones running around this place, particularly at dusk or at nighttime. It would be very dangerous. And so if hominins are coming in, say at night to collect ducks when they're kind of more tied to their nests, it might be an easier time to do it. They might be having these little task groups going in and doing that sort of thing, or another time coming in to scavenge larger animals like rhinos, but you have to scare off other predators. So you have to avoid predators or somehow drive away other scavengers. It's not an easy thing to have this area as your main area for collecting your food and water and plant resources and so on. So I think all of this tells us really that so if we take the protein residue results, we take our zooms results, we take our faunal remains, our stone tools, and so on, and we put them in the larger landscape of where we know there are other sites, probably sites where these people were living, rather than sort of coming in more opportunistically, whereas that we think that's what they're doing at Shushan Marsh. It really shows us that at 300,000 years ago, these individuals were much more sophisticated technologically, socially, and cognitively than archaeologists ever realized before this research was published. And I just wanted to show you some of the tools here. These are photos taken by me and Dan. So we have a Levantine blade that was positive for rhino, a scraper for bovid. So again, some kind of wild cattle. And then another point for camel. We have horse, or as I say, probably some kind of steppe ass. And then on the other side, duck. So just in conclusion then, using the lithic residue, the lithic data, the residue data, the faunal and paleoenvironmental data, we were able to combine these together to reconstruct an idea of hominin lifeways in a desert oasis during a period of increasing aridization. What we can see is that adapting to these local pockets of very concentrated resources throughout what is generally a very, as I was trying to show in some of my slides, a very resource poor landscape, was likely critical to the success and the survival of these early hominine populations. And again, this ability to be able to know where the next oasis area was and to be able to have that kind of mental map of their surroundings and when they should be where in time. And so, as I also mentioned, non-human predators would have also been drawn to the oasis, especially when you have these large carcasses exposed during butchering events. And so these hominine groups must have had well-established strategies or mechanisms in place for competing with or avoiding these other predatory species. And so this realization, combined with the need to maintain high mobility across areas of very poor resources throughout the oasis, provides sort of, as I say, a glimpse into those social dynamics that they would have had to engage in. And it really tells us why they were so successful in these arid environments. And so these behavioral strategies then were fundamental to the successful dispersal of these late Middle Pleistocene populations throughout Eurasia. And as I say, everything we can see suggests that these humans were more technologically, socially, and cognitively sophisticated than we previously realized. So I would just very briefly like to thank the Social Science and Humanities Research Council of Canada and the Leakey Foundation for generously funding our ongoing research, the Department of Antiquities in Jordan, the American Center for Oriental Research in Amman, the Azraq Wetland Reserve, and the Royal Society for the Conservation of Nature, for allowing us to do our research and facilitating that research. We have a number of colleagues, and again, I'd like to do a shout out to John Fagan, who's here tonight, and Cam, who isn't, Cam Walker, and of course Dan, who of course is here and here this evening, and then finally to the people of Azraq. I would like to thank them for sharing their rich cultural heritage with us. So shukran to all of them. Thank you to all of them and to all of you here tonight. And that's just our group. On the left is a very old picture of when we first started in 2012. And there on the right, you see a picture of Dan. I didn't know whether to have Dan in his archaeology mode or his drumming mode. So I chose his teaching archaeology mode. And then of course, there's Cam in the corner at the bottom there. Thank you very much.
April, thank you very much. That was absolutely wonderful. I'm going to start it off with a question, and then we'll figure out how to get the other questions. Okay.
Okay.
So my question is, you made a point in that last slide of saying that folks weren't living at Shishan Marsh because of the predators and all the rest of that. They seem to have left a lot of tools behind at a place they weren't living, number one. And second part of that question, have you found the living sites where they were living? Are there rock shelters in the vicinity or something like that?
Yeah, so a lot of the, as I say, a lot of the you know, when I say tools, I need to say that in a very broad sense. So a lot of that is chipping debris. So so we know that they're making a lot of tools on site, and you know, some of them they're using, it looks like, just very briefly and then discarding them. And you know, except for the hand axes that seem to be a bit more curated, they're doing a lot of, it seems, on the spot stone tool making, and so that kind of leads to this buildup of debris. We're also talking about a period that's almost 200,000 years old, you know, in length. So you know, that we have sort of a palimpsest in some ways, like a buildup of that material. And then the second part of your question, have we found where they're living? Yeah, we have actually, we haven't done a lot of excavating in the region around, but we have identified, I think, and I'm trying to remember what we're up to now, about 13 or 14 other sites that are just within, as you know, going to say a stone's throw. That seemed like a bad pun, so I won't say that, but it kind of is true. Really close by. And so in some other archaeologists working sort of in, about 25 to 50 years ago have also identified a number of other sites in the region. So we think they're probably living in some of these other ones and then coming in for what they needed from Shishan Marsh and going back to these slightly safer areas.
Okay, great. Thank you very much.
Yeah, you're welcome.
Julia, how do we work this question thing?
Should I look at the chat or how would you like to do it, Julia?
The thing in the chat, the main question I see is from Terry Dolan. So there is enough residue that all kinds of species sera can be tried to rule out the source of the residue?
So do you mean, is there enough residue on the stone tools themselves?
Yeah, I think the question is how there can be so much residue that you can test out all the different sera.
Yeah, so and John can probably pop on and tell you exactly the amount that you need to do the testing, but certainly, yeah, we've had no trouble. We've tested against, and I'm trying to remember, at least 10 different sera, and some we did 10 different animals just to be able to rule out some of them. So we did goat, for instance, because there's goat all over our site. So we wanted to make sure we weren't getting any contamination from modern fauna, and we don't have any goat, for instance. We don't have any goat positive for goat on any of our things. So yeah, we definitely have enough residue, and we're able to freeze that residue and then test it when we come up with new ideas or more money to do further testing in the future. And in fact, we did retest some of them recently, which was quite, stuff that we had collected quite a long time ago, and we were still getting the same results. And so that's been actually an interesting kind of methodological thing that we've found works, which we didn't know necessarily before. So that's the long answer to yeah, we definitely have enough.
Thanks, that's great. Are there any questions here from the audience? I think we can bring a microphone.
If you had found any reptile or amphibian remains because of the marshes?
No, we haven't actually looked for those yet, and that would be kind of interesting. Yeah, I have like future ideas about what to do on the. One of the things I would like to do is expand. We did do some testing more broadly for birds, for instance, but we haven't. But it's a little bit more complicated. But it's something that I'd like to look into a bit more in the future. But in terms of reptiles and amphibians, we did want to test for turtle. That was one of the species types that we got funding for from the Leakey Foundation. Unfortunately, there was commercially available turtle antiserum for a short time, and we didn't grab it, and it hasn't appeared since. And we don't want to do it custom. We don't want to do it in-house because it's the method for getting it is pretty terrible. Apparently, you have to drill through the turtle. Sorry, and I haven't even started drinking yet. So the turtle or tortoise shell, which sounds horrifying. So we're not doing that. So we're trying to, as I say, we try to do it when there's some kind of veterinary procedure that's necessary for a blood draw anyways. And so we've been trying to work with a bunch of zoos. I've emailed and called turtle rescue places and so on, and we just haven't been able to do that yet. And we haven't tried for amphibians. We were thinking about fish, but there's only one fish that would have been there at that time, and it's so tiny that it really doesn't make sense. Like they wouldn't have been using a tool to cut it up, at least according to our ZooArc person. So again, that's the long answer for no, but the broader answer is that I don't think we're finished. doing the kind of research that we want to do. We just have to apply for more funding and expand our inquiry.
All right. Online, I see another question. Have you found any human footprints near the site?
No, we haven't. I really wish we had. There are literally thousands of footprints that date to the Paleolithic, and they go all the way back to Australopithecus afarensis, and there are lots of footprints associated with Neanderthals. And pardon me, and because one of my areas of specialty is the archaeology of children in the Ice Age, I've been looking a lot at children's footprints. And so we have a lot of children's footprints that date to the Paleolithic, because as probably all of you know, kids often play in unexpected areas. So their footprints are not obliterated by later foot traffic. So, you know, the way some adults would be. So we find evidence in France, for instance, down along the border with Spain. There are caves there where we have evidence of children playing, you know, sort of making the perfect footprint or running around with other kids. And we have little mud ball pellets that they've thrown at each other and at stalagmites and so on, and that kind of thing. So that gives you so much information, you know, height, sex, sometimes anyways, age, speed of locomotion, all kinds of things, whether they're wearing shoes or not. So I wish we had that at the marsh, but we really don't have that level of preservation. Unfortunately, you know, no one has ever uncovered any there, but that would be the dream for sure. Well, the dream would be to have hominine fossils. This would be second to that dream. And they're all over, you know, I won't say all over, but they're in Israel, so they're nearby. So I feel we must have them. We're actually excavating another site in the town called Madaba, which is maybe an hour or so away. There we have a giant sinkhole about 25 meters or 75 feet in depth, and we have incredible faunal preservation there. It's just so dangerous that it's you know, to get a drill in there to take soil samples and so on, or to be able to, Jim Pukinis, our zooarch person, is repelled down that. And so we've found some really good faunal remains. We're really hoping that like an ancient hominin tripped and fell into this, or you know, as Jim always shows this slide of someone being pushed into.
Sort of like Cava de los Oasis in Spain, right? Where you putting
And I wish
people in
we
there. Cool.
Exactly. So we have that kind of site, but we've only been able to get back a few thousand years, and we have to go back with better equipment and more funding to be able to do it safely. So you know, that's a stay tuned as well, I guess.
So have we got any other questions from the audience?
Or Dan, was there anything that you wanted to add to anything I missed about the tools that you wanted to add?
Well, gee, April, no, it was great talk. And other than, yeah, you know, we need to finish that paper. Yes. I'll be up as soon as possible. And no, it was, you know, an honor for me to get to work with April on these tools for a long time. And they are spectacular. The hand axes are literally, you could butcher an animal today with those hand axes that are 200,000 years old. That's how sharp they still are. It's extraordinary. So yeah, it's been a fun project. And to add, nothing except April Nowell's pretty awesome.
Question?
My mom pays him.
Royally.
That's right.
Have you created serums for Arabian horses versus donkeys to see which line of equus that's for?
John, do you want to talk about that since you're here? Do you want to talk about the different? My understanding is that we just tested for a horse more broadly as opposed to
Yes.
Specific species or anything like that. And then what we do is then we look at what we know is around in that region at that time based on other sites and other work and so on. And we make a logical guess that way.
And any of the equus or horses would be ones that would only have been around in the Pleistocene. So they wouldn't have been like Arabian horses yet, or what we know from modern horses. They would be, like April mentioned before, different asses or mules, so to speak, or Pleistocene horses, of which we know there's some extant ones in Mongolia still, but those kinds of early types of horses are, and the same thing with all the species. There were Pleistocene versions of the modern ones. What we can use is residues from the modern ones because it's the same species family. And so, we can, that's how it all works. But, it's not as clear cut as, when we have residue for the duck residue, it could be maybe a goose. We just don't know, same family.
Mm-hmm. And again, coming back to what's likely in the region and so on helps us narrow that down. Someone asked if we found coprolites. No, unfortunately not. And then stone tool materials that came from outside the local region, other than Flint, were you not? So they're using those local wadi pebbles or local valley pebbles, and then Flint from those farther away sources. That's what we have our stone tools.
We're gonna have to call it off here in a couple of minutes. It's 8:30.
Oh gosh, right.
Yeah, it's about 8:30. We've got a couple of quick questions. Maybe you can just answer these in a kind of a yes or no thing. One person has asked, did you find any evidence of? Oh, she did?
Yeah.
Okay, great. Done. Sorry, I was looking at my watch and trying to go, well, we got to get out of here because we've only got it rented for so long.
Oh, yikes. Okay.
Okay, April, thank you very much. We appreciate the talk, and everybody learned a lot, at least I did. So great. We really enjoyed the lecture. Thank you very much.
Thank you so much for having me. It was super fun. It was a real pleasure to be here. And I'll wish you all a nice evening. And I'll talk to you later, Dan.
Talk to you later, April.
Okay, take care, you guys. Good night. Bye-bye.
On your way out, you can walk by the magazines and pick some up if you so desire.